Our next portion in this lecture will cover the physiology of digestion and absorption. Digestion breaks down ingested foods into their chemical building blocks. Only these broken down molecules are small enough to be absorbed across the wall of the small intestine. Digestion is a catabolic process that breaks macromolecules down into monomers small enough for absorption. Intrinsic and accessory gland enzymes are involved in digestion. Enzymes carry out hydrolysis, where water is added to break chemical bonds. Absorption is a process of moving substances from the gut's lumen into the body. Tight junctions ensure molecules must pass through epithelial cells rather than between them. Materials enter the cell through the apical membrane on the lumen side and exit through the basolateral membrane on the blood side. Lipid molecules can be absorbed passively through the membrane, but other polar molecules are reabsorbed by active transport. Most nutrients are absorbed before the chyme reaches the ileum. Starting with carbohydrates. Only carbohydrates broken down to monosaccharides can be absorbed. Starch and disaccharides are broken down to oligosaccharides and disaccharides, beginning with salivary amylase in the mouth. They are further broken down into lactose, maltose, and sucrose. The final breakdown results in the monosaccharides of glucose, fructose, and galactose. Figure 23.34 is a great figure to study. In your text, there is a similar figure for each of the macromolecules, and you will need to know what enzymes are produced, from where, that allow the absorption of each macromolecule. Carbohydrates are broken down first in the mouth by salivary amylase, then in the small intestine by the pancreatic amylase, and lastly by the small intestine brush border enzymes dextrinase, glucoamylase, lactase, maltase, and sucrase. Polysaccharides are broken down to monosaccharides for absorption. The steps of starch digestion are as follows. Pancreatic amylase breaks down starch or glycogen that escape the salivary amylase into oligosaccharides and disaccharides. Brush border enzymes dextrinase, lactase, glucoamylase, maltase, and sucrase further break down these into the disaccharides lactose, maltose, and sucrose, and finally into monosaccharides like glucose, fructose, and galactose. The monosaccharides are co-transported across the apical membrane of absorptive epithelial cells. This active transport uses the sodium concentration gradient established by the sodium-potassium pump in the basolateral membrane. Monosaccharides exit across the basolateral membrane by facilitated diffusion and enter the capillary via intracellular clefts. People with lactose intolerance have deficient amounts of lactase and cannot consume lactose. Any lactose eaten remains undigested and creates an osmotic gradient in the intestine that prevents water from being absorbed, resulting in diarrhea. Moving on to the digestion of proteins. Proteins are not only digested, but there are digestive enzymes and proteins from the breakdown of mucosal cells. So that means we have a lot of other proteins in our digestive tract that could be possibly digested, so we have to be careful. Proteins are broken down into large polypeptides, then small polypeptides, then small peptides, and finally into amino acid monomers with some dipeptides and tripeptides. Digestion begins in the stomach, where pepsinogen is converted to pepsin at a pH of 1.5 to 2.5. Pepsin becomes active in the high pH of the duodenum, becomes inactive in the high pH of the duodenum. Proteins are broken down into large polypeptides by pepsin, produced and occurring in the stomach. In the small intestine, pancreatic trypsin, chymotrypsin, and carboxypeptidase digest large polypeptides to small polypeptides and peptides. In the small intestine, brush border enzymes like aminopeptidase, carboxypeptidase, and dipeptidase further break the peptides down into amino acids. This figure illustrates the digestion and absorption of proteins in the small intestine. First, pancreatic proteases break down proteins into small pieces and some individual amino acids. Next, brush border enzymes break the protein fragments into amino acids. Thirdly, amino acids are co-transported across the apical membrane of the absorptive epithelial cell. This active transport uses the sodium concentration gradient established by the sodium-potassium pump in the basolateral membrane. Lastly, amino acids exit across the basolateral membrane via facilitated diffusion and enter the capillary via intracellular clefts. So the absorptive process of amino acids in the small intestine is the same as the monosaccharides. In rare cases, intact proteins are taken up by the intestinal epithelial cells by endocytosis and are released into the body. This is most common in newborns because of the immaturity of the intestinal mucosa. This may result in food allergies as the immune system sees the intact protein as antigenic and mounts an attack. Allergies usually disappear as the mucosa matures. I'm going to go past this. We're going to cover that in just 1 second. Oops. All right. Now let's move on to fats. So lingual lipases in the mouth and the gastric lipase in the stomach are of minor importance when it comes to fat digestion. So these two are of minor importance. Fats require emulsification because triglycerides and their breakdown products are insoluble in water. So, fats require a pretreatment with bile salts produced in the liver and delivered to the small intestine to break down the large fat globules into small ones. Then, pancreatic lipases can work down on these small fat globules. They break down the fat into monoglyceride plus 2 free fatty acids in the small intestine. Next, there is a mycelial formation, where the products from fat digestion become coated with bile salts and lecithin. Absorption occurs when lipid products leave the micelles and cross the epithelial membrane via diffusion. Then chylomicron formation occurs, when lipid products are converted back to triglycerides and packaged in lecithin and lipoproteins, forming the chylomicron. Chylomicrons are exocytosed from the basolateral side and enter the lymphatic lacteal. This eventually is emptied into the venous blood at the thoracic duct. Once in the blood, chylomicrons are broken into free fatty acids and glycerol by the lipoprotein lipase, so they can be used by the cells. Some short-chain fatty acids can diffuse directly into the blood and then be transported to the liver via the hepatic portal vein. This figure illustrates the emulsification, digestion, and absorption of fats. First, emulsification of fat is necessary. So bile salts in the duodenum break the large fat globules into small fat droplets, increasing the surface area available for lipase enzymes. Next, digestion occurs when pancreatic lipases hydrolyze the triglycerides, yielding monoglycerides and free fatty acids. Next, micelle formation occurs when micelles ferry their contents of fatty acids, monoglycerides, and bile salts to the epithelial cells. Fourthly, fatty acids and monoglycerides diffuse from the micelles into the epithelial cells. Fatty acids and monoglycerides are recombined and packaged with other fatty acids and proteins to form chylomicrons. Lastly, chylomicrons are extruded from the epithelial cells by exocytosis. They enter the lacteals and are carried away from the intestines in the lymph. Our last macromolecule to discuss is the digestion of nucleic acids. Where do we find nucleic acids? How do we ingest them? Well, the nuclei of ingested cells of food cells contain DNA and RNA. so they must be broken down. Pancreatic nucleases hydrolyze nucleic acids to nucleotide monomers. Brush border enzymes called nucleosidases and phosphatases break nucleotides down into free nitrogenous bases, pentose sugars, and phosphate ions. Breakdown products are actively transported by special carriers in the epithelium of the villi. Nucleic acids are broken down by pancreatic ribonuclease and deoxyribonuclease in the small intestine, and then further by the brush border enzymes nucleosidases and phosphatases, also in the small intestine. The digestion products enter the intestinal cells by active transport via the membrane carriers. They are then absorbed into the capillary blood in the villi and transported to the liver via the hepatic portal vein. Digestion also includes the absorption of vitamins. This occurs in the small intestine. Fat-soluble vitamins A, D, E, and K are carried by micelles or diffused into the absorptive cells. Water-soluble vitamins C and B are absorbed by diffusion or by passive or active transporters. Vitamin B12, a large charged molecule, must bind with intrinsic factor and then is absorbed by endocytosis. Oops, what happened? In the large intestine, vitamin K and B vitamins from bacterial metabolism are absorbed. Electrolytes must also be absorbed. Most ions are transported actively along the length of the small intestine. Iron and calcium are absorbed in the duodenum. Sodium absorption is coupled with active absorption of glucose and amino acids. Chloride is transported actively, and potassium diffuses in response to the osmotic gradients. It is lost if water absorption is poor. Iron and calcium absorption is related to their needs. Ionic iron is stored in mucosal cells with ferritin. When iron is needed elsewhere, it is transported in the blood by transferrin. Calcium absorption is regulated by vitamin D and the parathyroid hormone. Water absorption is also critical for our survival. 9 liters of water, mostly from GI tract secretions, enter the small intestine. 95% of that is absorbed in the small intestine by osmosis. The rest is absorbed in the large intestine. Net osmosis occurs if the concentration gradient is established by active transport of the solutes. Water uptake is coupled with solute uptake. Malabsorption can be caused by anything that interferes with the delivery of bile or pancreatic juice. Damaged intestinal mucosa from a bacterial infection or some antibiotics are a couple of causes of malabsorption. Gluten-sensitive enteropathy or celiac disease is a common malabsorptive disease. The patient has an immune reaction to gluten. Gluten then causes damage to the immune cells in the intestinal villi and brush border. The patient is instructed to eliminate gluten from their diet. The GI tract activity declines with age. It produces less digestive juice, absorption is less efficient, peristalsis is slower, all causing a decline in the frequency of bowel movements. Smell and taste become less acute, and periodontal disease often develops. Diverticulosis, fecal incontinence, and GI tract cancer are also very common. Stomach and colon cancers rarely have early signs or symptoms, so metastasized colon cancers frequently cause secondary liver cancer. Oral cancers can be detected during dental exams. Most GI cancers are treatable if caught early. Therefore, regular dental and medical exams can prevent these cancers.